3 lectures on carbohydrates, 3 on lipids, probably 2 on proteins, and then the last 2 will be kind of different topics that we'll talk about closer to. So today we're going to talk about carbohydrates. So we've already talked about carbohydrates from the respect of digestion. So we've already talked about the use of amylase and how amylase is able to break down branched and linear carbohydrates that contain alpha-1,4, alpha-1,6 bonds. So amylose and amylopectin. What we're going to talk about today is a little bit more of the structure and the chemistry of these systems, as well as the flavor perception. So when we talk about the molecular structure of carbohydrates, they contain carbon, hydrogen, and oxygen only. So carbohydrates could differ depending on the number of... This is a weird view I have. Do you have a good view? Yeah. Huh. So they're organic molecules that have carbon, oxygen, and hydrogen. These are the most abundant macronutrient found in foods. When we talk about carbohydrates, it's important that we're able to delineate and differentiate the different monosaccharides. So the first thing we look at when we talk about the monosaccharides in the food industry are whether or not the oxidized group is in a ketone position or an aldehyde position. I'm just going to see if I can fix something. This is just a really... There we go. Okay. So in the case of an aldose sugar, where the carbonyl group is an aldehyde, we get... Where'd it go? What is going on here? My slides are all messed. Just one second. I see what the problem is. There we go. So we have monosaccharides. Again, those are simple sugars. Monosaccharides include things like glucose. Why is that pointer? There it is. So glucose. Disaccharides are 2 monosaccharides covalently linked together. Oleosaccharides are anywhere from 3 to 10 carbohydrates, and polysaccharides are over 10. The defining features of these are dependent on the molecular weight. So typically when we talk about sweeteners, we talk about anything less than a disaccharide. So maltose, lactose, and then the monosaccharides, glucose, galactose. Oleosaccharides don't contain any sweetness. So these don't provide a flavor attribute to that food. Nor do they provide any kind of viscosity enhancing properties. So when we talk about things like pectin, which you make jello out of, or sorry, jam out of, or alginates, the pimento inside an olive. So you know the olives, when they take the pit out, that green or the red little pepper inside, that's an alginate gel, which we'll talk about. So polysaccharides don't provide sweetness either, but they have a lot of unique functional properties in structuring water. So think about a starch gel, gluten. All of these different things are associated with the long molecular weights or the long chain length of those polysaccharides. So when we talk about the structure of sugars, oftentimes the importance gets lost because we're talking about conformers, diastereomers, enantiomers, which we'll talk about in a second. But it's extremely important that we're able to, again, differentiate based on the chemical structure, the type of sugar you have. It's not overly important that you know how to name them, but it's important to understand that the minor structural changes have extremely important consequences, not only in the sensory perception, but also the functionality of those sugars and carbohydrates. So again, when we talk about enantiomers, so when we talk about sugars, there is 6 carbons typically for most sugars we talk about, or 5, but typically 6. The most reduced one, if it's an aldose sugar, is located at the top of the chain. Now, remember Fisher projections from first year chemistry? The ones going vertical are in plane, right? So they're going into the board. The ones horizontal come out of plane, right? So depending on which way those hydroxyl groups come out of if they're on the right or the left, determine whether they're a D or an L isomer, right? Does anyone remember what D and L stand for? D is yeah. Yes, so what does D stand for? Like, you're right, it's on the left side. It is dextrorotary. L is levorotary. So what is that telling you? What is dextro and levo? Yes?
It's telling you like the orientation of like hydrocarbons.
Okay, and what physical manifestation does that have? Anyone remember how you differentiate if you have a D or L isomer? You probably, yeah?
You look at the hydrocarbon furthest away from the bottom group.
Okay, so let's pretend we don't have the structure. So we have a solution of carbohydrate. How do we tell if it's D or L? Remember the way it rotates light. So dextrorotary, levorotary. Rotates left or right. So depending on if you're dextrorotary or levorotary, the physical properties of that changes. Now, many of you probably don't think that's overly important. Here we have two spices. This is caraway from Bulk Barn, and this is spearmint. The main volatile component of each of these, so when you smell them, they are enantiomers of one another. So the smell difference between these two molecules is simply the relative position of a carbon, or sorry, of a hydrogen on a chiral carbon. Sorry, I just bought these this morning. I didn't open them. So, again, oh, it just went up my nose. Oh, that was horrible. Could you imagine if it was pepper? Oh, that was terrible. Don't put your nose too close. Oh. So again, just the chirality of the position of these two compounds drastically change the aromatic perception.
So they smell of physical property because like for enantiomers they should have like the exact same chemical property.
Right. Great question. We really don't understand how got to be careful how I say this. How a molecule interacts with an olfactory cell, we don't understand the mechanism for why that gives such a different perception. For example, oh, that burns so much. In the case of, I'm going to sneeze now, and it's going to be stuck. Oh. Okay. So in the case of sweetness, we barely understand how kind of the molecular reception of that molecule induces sweetness, which we'll talk about, but we don't understand how that molecule forms a circuit to trigger a neural impulse. It's beyond what we know. Does anyone else, you guys might be too young, does anyone remember a drug recall that happened because a different enantiomer was created on the industrial process of the drug? Thalidomide. So what was thalidomide used for?
To help prevent them from falling asleep.
To prevent them from getting morning sickness. So what's interesting about thalidomide is completely safe. And it has very, very good benefits in preventing nausea. The problem was when they scaled up the manufacturing process of it, they got both enantiomers and they didn't separate out the other enantiomer. And the other enantiomer was teratogenic, meaning it caused birth defects. So when if you were exposed to if a developing fetus was exposed to thalidomide, they developed improperly formed limbs that resembled, and the term flipper babies came from thalidomide. So actually, if you go now and ever become pregnant again, thalidomide is back on the market as a way to prevent nausea. They just now know that they can't have a racemic mixture of those two compounds. So understanding that simply which side a hydroxyl group is on has very, very important biological consequences. Reactivity of those molecules change. Site receptors, if they interact with proteins or enzymes, will change. So when we talk about configuration, the configuration of a sugar is determined at time of synthesis. The configuration is if it's a D or an L configuration of that sugar. Configuration does not change after it has been synthesized. So configuration is fixed. When we talk about different configurations, an enantiomer is an exact mirror image of another molecule. So if two molecules are enantiomers, what that means is here you see you have an S configuration, here you have an R configuration. Where you have an R configuration here, it's an S configuration here. So all of them are opposites. So they're mirror images. In a diastereomer, they are not mirror images, but the hydroxyl groups are not on the same side. So if you look here, we have an S in the first one. Here we have an R. Then we have an R. But now we have an S, here we have an S, here we have an R. So it's not a mirror image, and at least one of those chiral carbons are of opposite configuration. So we have two different conformers. We have enantiomers, which are mirror images, and diastereomers, which are not mirror images. Does that make sense? So a diastereomer and an enantiomer are generally classified as stereoisomers, and stereoisomers are determined at point of synthesis. So when that plant is undergoing its biochemical reaction to synthesize its polysaccharides, that position or that chirality of that carbon is determined at point of synthesis and doesn't change when it's in its environment. Does that make sense? Now, configuration differs from conformation. Conformation is dependent on the environment in which it is in. So if you take a sugar solution and put it into an aqueous environment, conformation will remain the same. Sorry, configuration will remain the same. Conformation will change. So when we draw a Fischer projection of a sugar molecule, so how many of you taken biochemistry? So when we talk about this structure of a sugar, is it in high prevalence in the environment? Is this how sugars naturally exist in solution? No. What do? They become cyclic. They cyclize. Now, when they undergo that cyclization or they form that cyclic structure, that ring size, if it's a 5-carbon ring or a 6-carbon ring, is dependent on where that oxidized group is. So remember, we can either have aldose or ketose sugars, right? Aldose meaning it's at position 1, ketose meaning it's at position 2. So when it's at position 1 and it undergoes cyclization, it forms a 6-carbon ring. When we have a ketose sugar, it forms a 5-carbon ring. Does anyone remember how the strain of those bonds differs between a 5 and a 6-carbon ring? Where is there more strain in those bonds? In a 5-carbon ring or a 6-carbon ring? 5-carbon. So the likelihood of a 5-carbon ring going into its linear form is higher than for a 6-carbon ring because there's more ring strain, right? There's more energy that's required when it forms that cyclic structure. So when we talk about sugar chemistry, the vast majority of the time, 99% of the time, the sugar is going to be in this cyclic structure. So this cyclic structure, what's very important about it is when this hydroxyl group attacks this carbon here right now is non-chiral, right? So for a carbon to be chiral, it has to have 4 different constituents coming off of it. So we have a hydroxyl group, we have a carbonyl group, we have a long carbon chain, we have a hydrogen. So this carbon is chiral. This carbon is chiral. This carbon is chiral. This carbon is chiral. This carbon is achiral because it's got 2 hydrogens coming off. This carbon is not chiral. Now, when it undergoes nucleophilic attack, and that hydroxyl group and this carbonyl interact to form a cyclic structure, what happens is this bond breaks. We get a hydroxyl group up here and a hydrogen over here. That means that this non-chiral carbon, when it undergoes cyclization, becomes chiral. So that's referred to the anomeric carbon. Why this is important? One is because this will determine whether or not, when they form cyclic structures, is that cyclic structure able to open into its ring form or not? Does anyone remember what type of sugar is able to undergo a transformation in conformation between a cyclic and a linear structure? We have a really special name for those sugars. Talked about it on the first day of class. A reducing sugar. So reducing sugars have the ability to go back and forth between the cyclic structure and the linear structure. Why is this important? Because reducing sugars partake in chemical reactions known as the Maillard reaction, right? Non-reducing sugars, or ones that are unable to go back into that linear form, are unable to partake in the Maillard reaction. So when we talk about roasting of coffee, toasting of bread, the formation of that crust layer, all of those chemical reactions are the Maillard reaction, which are very, very important for imparting flavor and physical appearance of that material. So if it's brown, again, another one being maple syrup. Now, conformation is dependent on both the pH and temperature of that environment. So the higher the temperature, the more likely it is to go into that linear form. And pH, it is also dependent because we have to protonate that once it goes into the cyclic form. Sorry, into the linear form. So, it is extremely important that you're able to identify, based on the chemical structure, whether or not a sugar is a reducing or a non-reducing sugar. So on here, we have D-glucose. So again, our oxidized group is at position 1. So we have an aldose sugar. Because it's at position 1, we're going to have a 6-carbon ring, or a 6-membered ring, with oxygen being in that. So we undergo a cyclization through a condensation reaction, so we lose a water. So our position 1 is attacked by our position 5. So again, position 5 hydroxyl group forms a cyclic bond with position 1. So position 5 forms a cyclic structure with the anomeric carbon at position 1. Now, when that happens, you can see that that carbon at position now 1, when it's cyclized, which was position, I'm just trying to remember, so position 1 was up here, so now we're at position 5. So this was carbon 5 before. We have the hydroxyl group from position 5 coming over. It's now formed a covalent bond. I always get this mixed up. I hate doing it from the screen. So again, position 5 coming across, donating its hydroxyl group up to position 1. So here's the hydroxyl group at position 5, forming that 6th point in the 6-membered ring. If we go 1 over to this carbon, we notice there's no hydroxyl group coming off, right? Off position 5. If we go across to position 1, there is a hydroxyl group. So if that oxygen in that ring here is next to a carbon that has a free hydroxyl group, it is able to undergo cyclic to linear transformation. So again, we go to the oxygen in the ring. So there's always going to be an oxygen in the ring of the sugar. It doesn't matter if it's a ketose or an aldose sugar. If we look at the carbons one away, so either at position 1 or 5, there has to be a hydroxyl group there. Position 5, there is no hydroxyl group, so this will not open up here, but there's a hydroxyl group at position 1. So depending on the environmental conditions, this is a reducing sugar because one carbon away from that oxygen within that ring structure, there's a hydroxyl group. That's all you need to know to identify if it's a reducing sugar or not. Is that clear? So again, we have our oxygen group in our ring structure. We have a carbon here, we have a carbon here. One of those two carbons must contain a free hydroxyl group to undergo the opening reaction. So because this has a hydroxyl group, so again, if we look here, because this has a free hydroxyl group, there we go. Because this has a free hydroxyl group, this is able to undergo reversibly the formation of a cyclic structure or a ring structure. So that is the whole concept of a reducing sugar. Very, very important when we talk about the chemistry of sugars, because the pathways for degradation and flavor formation is dependent on the ability of that covalent bond to break and to reform, either being a linear reducing sugar or a cyclic non-reducing sugar. So we're going to come back to this. So depending on the conformation of the sugar, it can either go back and forth being a, again, reducing sugar, being able to go back and forth between the linear and the cyclic structure. If that hydroxyl group is gone or has partaken in another chemical reaction, it no longer becomes a reducing sugar. So where would that hydroxyl group be used? In what chemical reaction? Yeah?
Like condensation reactions, linking with other of the same unit.
So in condensation reactions is right. What is the most common condensation reaction in simple sugars? The formation of a disaccharide. So this hydroxyl group is essential in forming a disaccharide. So that anomeric carbon, which undergoes cyclization, is essential when we talk about the formation of disaccharides and then consequently in polysaccharides. So again, when we talk about our anomeric carbon of glucose, if we have the formation of, what do we have here? I think maltose, alpha maltose. So when we have the formation of maltose, we can see that this alpha carbon, this anomeric carbon here, is going to undergo a reaction with position 4 of another D-glucose. So when we have the 1,4 condensation, over here, this double bond, this covalent bond, is unable to become a reducing sugar. That hydroxyl group is now gone. It's now part of that covalent bond that forms those two sugars. So that side of that molecule is no longer reducing. This glucose molecule now becomes a non-reducing sugar, that half. Now, if we go to the other side of the molecule, does it still fit the definition of a reducing sugar? Yes. So maltose is still a reducing sugar. Even though the glucose, even though the glucose, the first glucose here is a non-reducing sugar, it still has a reducing component of that molecule. So maltose is still a reducing sugar. So 1,4 alpha maltose is a reducing sugar. If we had an alpha 1,1, that would no longer be a reducing sugar. So if we took this glucose and we flipped it over and now we took this hydroxyl group to react with this hydroxyl group, it would no longer be a reducing sugar. So as long as there's a hydroxyl group, one position away from the oxygen in the ring structure, anywhere in that molecule, it's considered a reducing sugar. So in the case of starch or amylose, amylose is just a whole bunch of these covalently linked together. That first glucose will still be a reducing sugar. So amylose still has a reducing potential. So when we talk about naming, obviously I'm not going to make you memorize naming in this course. If you take third year food chemistry, you'll have to know a little bit more about naming. But it seems like something that's so trivial to get you to memorize. But by understanding the naming, you can identify whether or not that's a reducing sugar. You can identify what type of bond it is between disaccharides, and why is that important? Why does it matter if it's a 1,4 or a 1,6 or a 1,3? Why does that matter?
You determine like how strong this disaccharide is when it's like linked to the same disaccharide. What
do you mean how strong it is?
So for example, let's say you have like two like beta
Anomers, yeah.
Yeah, so they would form a different glycosidic linkage, and then this could lead to like cross-linking to make it like a really rigid structure, right? It's like two beta.
Okay.
Make like cellulose.
Yes. Okay. So if it makes cellulose bios, what is that then? Can we break that down? We can't digest it. Yeah. So by knowing the naming, it will tell you whether or not the enzymes in our body can interact with it. Or, more importantly, in the case of things like non-digestible fibers, whether or not a bacterial species that's colonized our gut is able to break it down and ferment it. So, although it's outside of the scope of this course, it's still extremely important to understand how these molecules behave. Now, what's really interesting is all monosaccharides don't have the same intensity of sweetness. And disaccharides are less sweet, typically, than monosaccharides. But it's dependent on the configuration and the conformation. So think about this. Where those hydroxyl groups are in three-dimensional space determine the relative sweetness of that molecule. So if you have a conformation where that hydroxyl group is not in an ideal position, it won't be as sweet. So for some reason, we compare everything to sucrose. That's just the way we do sweetness profiling. So sweetness is dependent on the three-dimensional shape of that molecule. And that is highly dependent on the conformation and configuration of that sugar molecule. So we're going to talk about it in a second, but before we do, we have to talk about taste, and then we'll come back to that. I could literally spend the next week, or the rest of the semester, talking about taste. So it's really difficult to kind of break down what's important. And taste is extremely, extremely important. From an evolutionary perspective, we have evolved to prefer foods that are sweet. Why would that be? Don't worry about that we like it. The sensory perception, yeah.
Because sugar is like the most, like it's like a really simple carb, and we use a lot of carbs for energy, so it's easier to store it.
So it was used, right, it was used as a mechanism to select foods that are energy dense. So when you spend 90% of your time foraging, you want to minimize as much time as that as possible, so you identify foods that are calorie dense. You do that by identifying foods, typically, that are sweet. So we use it first as a way to identify nutrient-rich resources. We use it for something else. We use it to identify foods that have toxic compounds. So typically, we are adverse to consuming very bitter foods. Things that have a lot of caffeine. So coffee typically wouldn't have been a food of choice early on in an evolutionary path. So we use taste to make a determination whether or not that food is acceptable or unacceptable to consume. So taste plays an extremely important role, not only in evolution, but also today. So think about this. We have developed a taste preference for sweetness, which is also an identifier of nutrient-dense foods. In Western society, where very little of our time is spent foraging. I don't remember the last time I went into a forest or a field looking for food. So food is readily available. But we have been predetermined to select foods that are calorie dense, which is one of the reasons, or one of the theories behind one of the reasons why obesity is so prevalent. Because in our genetic makeup, at the very foundation of who we are, we desire foods that are dense in calories. Because again, from an evolutionary perspective, we have, for a very short path in our evolutionary history, for the last 200 years only, have we had access like we do to the foods that we have. Not only that, we now engineer foods to be sweeter and to be more calorie dense. And from an evolutionary perspective, we desire, we crave that. So it plays a very, very important role. Now, in high school, at least when I went through high school, we did different taste tests to say that we had different regions of perception within our tongue. We now know that's false. We know that the distribution of taste perception is not equal across the tongue, but we know that, for example, there's not a localized region that only senses bitter. How we now think of flavor is we have three main areas of perception. The front middle of our tongue, the back sides of our tongue, and the very back of our tongue. The taste buds don't differ drastically in what they sense. So taste buds in different locations can be more concentrated in sensing bitter, but they still have the ability to perceive sweetness. So within this taste bud, there are 4 types of cells. And those 4 types of cells will interact with different types of molecules to trigger neural impulses that detect a sensory perception. So the 5 basic tastes: sweet, sour, bitter, salty, and umami. Anyone know what the flavor sensation of umami is? Better yet, yes? Flavoring? Savory, yes. What ingredients are really good at promoting that savory flavor?
Like certain amino acids like glutamate.
Glutamate. So this is why if you go to Campbell's Soup and you buy their chicken broth, you'll see autolyzed yeast extract. They're trying to get those specific peptides that give umami flavors. Meats are really rich in umami flavor. There's also right now a big debate on what fat is. Do we taste fat? And it's this big debate on whether or not it's a mouthfeel, so the lubricity, does that change sensory perception? Or is there an actual receptor for fat? We don't know right now. So right now, literature's kind of back and forth on whether or not fat could be considered a 6th taste. What's really cool about the taste bud is different cells perceive different tastes. So our type 1 cells are very, very sensitive to salt. We can prove this by creating genetic knockouts of mice and then overloading their food with salt. And if they have the knockout, they become insensitive to that perception. So type 1 cells are very, very sensitive to perception of salt. Type 2 cells are really our taste receptors. There's all kinds of taste receptors. For sweet, for umami, so again, that's that mushroom taste, glutamate, MSG, ING. So a lot of Asian cuisine really relies on umami. And bitter. Type 3 cells, again, are very, very important. Whoops, jumped one. Are very, very important for determining sour. So the acidic compounds. And then type 4 are at the base of the taste bud, and these are just immature taste cells. So they're differentiating cells that eventually replace the taste bud cells that deplenish. Now, when I was putting this lecture together, my mind was boggled at this. When you look at type 2 taste receptors, what do you see on there that we've seen before? Leptin, neuropeptide Y, ghrelin, CCK, GLP-1. Where were these hormones that we talked about them? In satiety. So you actually have cells in your gastrointestinal tract that resemble taste buds, or that resemble the cells found in taste buds. So your gut is using the same type of cells that your mouth uses to detect sweetness, bitter, all along your GI tract. So when it detects sweetness, or when it detects a fatty acid, it can trigger the release of hormones to trigger satiety. But it's remarkable to think that our taste buds in our mouth have the ability to detect hormones that are produced upon consumption of food in our gut lining, in our large intestine, and even in the case of bacteria producing PYY. So how does, and there is no answer for this question, how does, or why does these taste receptors contain receptors for hormones that are essential in our gut for satiety and satiation? Is there a correlation? Is there a genetic reason, or is there a genetic predisposition based on the prevalence of receptors to lead to chronic diseases associated with diabetes? Again, we don't know, but there's evidence to suggest that there's a reason they're there. They're not just there by accident. The body doesn't produce receptors that are energetically not favorable to produce for no reason. So it's crazy to think that the taste perceptions in your mouth are what we use for chemosensation all along that GI tract. And again, that then influences the ileal brake. If that influences the ileal brake, that influences satiety. It also influences the rate of gastric emptying, which influences the rate of digestion. The rate of digestion then influences the glycemic index. All of this is interconnected. So everything from taste to gastric emptying, to satiation, satiety, to food form, all of these things are interconnected, and we can't talk about one without talking about the other elements. The abstract of this article, which I read again and again and again last night, really had an impression on me. So an improved comprehension of the relationship between taste, palatability, taste receptors, hedonic acquisition, so again these are the environmental and the social cues that influence eating, might lead to an increased understanding of the biological underpinnings associated with energy and consuming healthy amounts of energy. So again, when we talk about obesity, and we talk about the prevalence of obesity, we all know, everyone knows, that it's not healthy to be obese. I don't think anyone in the world would have a misconception that being obese is healthy. But why? Why do we still maintain a biological drive to eat? It's more than just hedonic. It's more than just, well, we're never feeling full, we're always in large groups when we eat dinner, so we get obese. Is there a genetic makeup? Again, those interactions of hormones and taste perception, are there biological underpinnings in determining whether or not we have a higher prevalence of becoming obese? The other thing too that we have to consider when we talk about taste is there is a whole class of sensory perceptions that don't rely on the typical sensors that we talk about, or the typical taste buds. Can anyone think of examples of those? So what are some other perceptions that we perceive in our mouth? One is temperature, which is from the, I can never say it, trigeminal nerve. But what else? What other perceptions do we perceive in our mouth?
Spicy.
So spicy or pungency, how hot that food is. So those molecules typically are non-polar. So if you ever do one of those challenges, I love watching them, where you buy the Grim Reaper chip, that single chip in a little coffin box that's like covered in 1,000 or 1,000,000 Scovilles of heat units. What are you supposed to drink when you eat something that's really hot? Milk, because it is fat, because it can solubilize capsaicin. What are some other ones? How many, yeah. Okay, texture. So that's all with the transgeminal nerve. So texture, not really a flavor, but yeah. Acidity, that's sour. So that's one of our 4 main ones. Who here drinks wine? What's the difference between red wine and white wine? Not color. Tannins. Yes. So what do tannins do? What is red wine? Why is red wine different than white? When you drink it, red wine causes puckering, astringency, right? So those tannins, they're able to interact with the proteins in your saliva, and they coagulate. And that gives you that feeling of dryness in your mouth, because you have this coated layer of proteins that are all coagulating onto your tongue. Another really important one that hasn't really made it into Western society yet, very, very, very important in Asian culture and food. If you go to China, Japan right now, and you look at all the beverages have it. Menthol, that cooling sensation, right? Very, very popular in Asian cuisine right now. So the biological perception of taste is extremely, extremely important. Okay. The concept of genetics playing a role in taste was first introduced in 1931, and it happened at DuPont Chemicals. There was this gentleman named Dr. Arthur Fox, and he was working with a chemical called PTC. So that's it there. And what he found was, they had a little bit of a lab accident that created basically an aerosol of PTC, and the staff behind him all started to get this really bitter taste in their mouth. But he was working with the aerosol, and he had no perception of it. And what he did then, without knowing if it was safe or not, he brought that chemical home, and he started feeding it to his wife, his friends, his kids, and he found that there was a very strong correlation based on lineage. So since he didn't taste it, his kids didn't taste it either. And what he found was, and which was really cool, was this depended on Mendelian genetics. So inherited genetics. So PTC, there is one gene responsible for the detection of, we say PTC, really what we're talking about is this CNS group. So this group of 3 compounds, or 3 compounds here, it doesn't really matter, is what's detected. Now, PTC is not found in foods. So there's no risk of consuming this in our actual food supply. However, this thioureal group, this NCS group, is found in all kinds of cruciferous vegetables. Broccoli, Brussels sprouts, very, very common. Now, before we do the taste test, I'm going to give a little warning. This compound, they now say, is carcinogenic. Having said that, a single exposure at the concentration on that strip of paper is no risk. So people do this experiment all the time in grade schools and in high schools. It's completely safe. If you have allergic reactions to sulfur-containing compounds, I wouldn't consume the PTC. But having said that, I did it with my kids last night. It's not something, it's chronic exposure that has shown any risk. Again, this is so dilute, and a single exposure is not going to create any risk. So what I want everyone to do is take the first strip, the control. There's nothing on this. This should give you no flavor perception. It's just a piece of paper. So everyone put that first strip in. Leave it so it soaks onto your tongue. Once you've gotten a good idea on how it tastes, yeah. Anyone have a gray box? Where are the gray boxes? Do you want to, are you guys still passing through? Anyone just have a set that they're not passing through still? Do the control first. Okay, they're still coming. Tastes like paper, yeah. Don't worry. How many people don't have it? Is it this whole back corner? Is there another container over here that's done being passed around? Where are the 3 containers? You're almost done?
No, you didn't. What is that? Okay, that's right. Okay. Put the lid on. What are you doing?
You guys don't have it either? You do? You guys don't. Okay, and there's one coming there too. So we should be okay in a minute. So everyone's done the first one. Pretty bland, right? No flavor. All right. Just to warn you, if you are a super taster, PTC is going to be extremely bitter. It's going to be gross. So if you are a super taster, and the reaction is going to be extremely different. When I did it, I had 0 perception. It was like the piece of paper in my mouth. My wife did it, and she gagged. What was that? All right. So let's go. Let's try PTC. %75% of the class should not be able to taste anything on PTC. So, and the reaction will be very different. You'll see people that put it in their mouth and instantly take it out. So who could taste it? Okay. So quite a few people. It's gross. All right. So if you tasted the second strip, you are what we consider a super taster. Now, what's interesting about that, if you tasted PTC, you are less likely to like foods such as cruciferous vegetables. So you don't like typically don't like things like broccoli, like Brussels sprouts. Those are very adverse foods for you. So of the super tasters who could taste PTC, how many of you like cruciferous vegetables? How many of you dislike cruciferous vegetables? Oh, so it's pretty split. Now, here's the other correlation they found. They found that the 75% of people who do not like PTC, who do not taste PTC, have an increased intake of high-fat foods. So people who didn't taste that bitter compound have a preference to consuming foods high in fat. So there's a genetic basis for food preference. And if there's a genetic basis for food preference, especially one as important of whether or not you eat cruciferous vegetables, really, really high in glucosinolates, right? Nutrition people should know glucosinolates are good for you. Now, if we take the thiourea next, this will determine whether or not you are a recessive or dominant. We'll come back to that in a second. This will determine whether or not you are a recessive or dominant super taster. So next, take thiourea. if you get, so thiourea, again, typically bitter. Not nearly as bad as PTC for the super tasters. Thiourea is way worse for you?
Like if we're going to take that
Now here's where it gets tricky. So sodium benzoate, the last one, what's interesting about it is you're all going to have different sensory perceptions to sodium benzoate. Some people will get sweetness, some people will get sour, and others will get bitter with the sodium benzoate. So you can go ahead and do that last one. So I can hear some people are saying, who gets a sweet perception from the last one? Sour? Bitter. So amazing, eh? The exact same molecule can differentiate so strongly. Anyone remember those little, they were plastic, I'll draw it. They were like a little plastic tub. And they were sealed with like a foil lid. And they had like a purple or an orange flavor. They were really inexpensive pre-juice boxes. So before the Tetra Pak or the juice box was invented, we used these little plastic containers that were that had a foil seal on them. The downfall to those was that packaging was really permeable to oxygen. So the color would go off really quickly. They would oxidize. So oxygen could diffuse through these, so they added sodium benzoate as a preservative. And this gives you, this when you were kids, and that weird taste you got from that juice was called benzoate burn. And I remember when we would have them in class, some kids, like for me, it just tasted like juice. Like I could only taste the orange juice or the grape juice. There were friends that I would give them to, and they couldn't drink it because they could detect that sodium benzoate. So it's remarkable to think that the chemical structure being identical, but the genetic variation in taste perception determines whether or not you can determine if that flavor is present or that molecule is present or not. And here's where it gets really crazy. There's research on it, and again, it's not as conclusive as whether or not it's a super taster or not, but it's dependent on where you were born, in what country you were born, what is your ethnicity. So for example, Inuit people have different scores on being super tasters than Caucasians. So it's genetically dependent on your upbringing, which is absolutely remarkable. So when I was at Rutgers University, there was a professor there named Beverly Hale. And what she studied was PTC, so the compound, that first one you determine, depending on how low of a concentration you can detect, determines whether or not you are a special class of super taster. So super taster has now been differentiated into many, many different segments. What this does, so now they use another compound called prop. I don't know what the difference is. Well, I do. That's prop there. That's PTC there. So again, the thiourea group is still present. There's the thiourea there. There's the thiourea there. So they're present in different foods, that basic moiety. So imagine again, your sensitivity to this thiourea determines your taste preference to that compound, or influences it. It's not the only determinant, but it plays a role in determining whether or not you prefer vegetables. As an adult, you can acquire a taste for bitter. You've all done it. If you have videos of when you first consumed chocolate, is a great example of an acquired taste. Nobody likes chocolate the first time they taste it. When you have kids, the first time you give your kid chocolate, videotape it. It's hilarious. The caffeinic acid, the different polyphenols that are in chocolate that make it healthy are what give it that bitter taste. Bitter is not always a preferred taste. Coffee is an acquired taste. You don't like the first cup of coffee you have, typically. Most people start drinking coffee by adding a lot of, and milk binds the bitter caffeine, the bitter compounds that are in coffee. So it doesn't taste as bitter. So if you're a coffee connoisseur and you put milk and sugar in your coffee, you are not a connoisseur. You just want the caffeine effect, which is fine. I only drink Tim Hortons. I am not a coffee connoisseur. But this is dependent on your prop status, on whether or not you are sensitive to thioureal groups. So again, if you think about it and you're genetically predisposed to have that adverse reaction, that bitter perception, when your mom and dad give you peas as a child, you're not going to want to eat them. If they give you broccoli, Brussels sprouts, the initial reaction of that infant is to spit them out. So how do you then train a population that is genetically disposed to not liking bitter foods to eat them because they're important? So this has become a fairly big thing in nutrition, is to understand whether or not your child is a bitter taster, because it totally changes intervention strategies if you have an obese child that's not eating coniferous vegetables. You have to introduce them slowly. How do you do that? By adding sauces and dips, right? You dip your broccoli in it to mask the flavor. If you're making fruit juices of grapefruit juice, you dilute it or you add sugar. So we can introduce the taste, just like we do with coffee by adding milk and sugar. We can train the body to not have that same reaction to bitter. So it changes whether or not we have a food preference. And that food preference can influence the status or the amount of energy we consume, and whether or not we're going to have chronic health effects based on our genetic profile of taste. It's not the only determinant, but it plays a role, which again is really remarkable to think about. So, genetic variability and the ability to detect bitterness can have chronic, long-term health effects associated with obesity. Children who are sensitive to bitter tastes have to be trained to like it. They have to be constantly exposed with different stimuli. And additionally, they have different markers in whether or not you're going to be preferential to high-fat diets. So again, when you talk about chronic obesity, it's not the cause, but it may play a role in. So getting back to that question on how does an olfactory sense detect the difference between a D and an L isomer? Again, we don't know the answer to that, but we have theories that are used to predict sweetness. We missed the tweet, don't worry about it. So, a sugar molecule, or for something to be perceived as sweet, has three characteristics. And these three characteristics are referred to the AHB theory of sweetness. So imagine you have a taste receptor. And just like a light switch, when there's nothing there, that switch is open. Right? So that switch is open, there's nothing there, there's no circuit or neural impulse that's transmitted. Right? When you close that switch, you put a bridge of metal across it, and that electricity can flow and the light comes on, or that neural impulse goes. Same thing as taste perception. The switch now being the molecule that then comes in and interacts with that site, creating a neural impulse. So in the case of sweet molecules, they have three functional groups. They have a group that can accept a hydrogen bond. They have a group that can donate a hydrogen bond. And then they have an aliphatic moiety that can undergo van der Waals interactions. And these distances between these three sites have to be 5.25 angstroms between our hydrogen bonding group. So our gamma moiety is our aliphatic, so this can be a long carbon chain, or it could be a phenyl group. We have our hydrogen bond, so our hydroxyl group, and we have our hydrogen bond accepting group. So in our taste receptor, we have this anchor group, which interacts via van der Waals interactions. This drives that molecule to the taste receptor. We then have our hydrogen bond group and our hydrogen bond accepting group forming two hydrogen bonds. So when that circuit is complete, it triggers a neural impulse. So imagine then you have this molecule, here's your taste receptor. Your hydroxyl group comes down, forms a hydrogen bond. The hydrogen bond receiving group comes and forms a bond. Now, if this hydroxyl group is flipped and going up, it doesn't complete that neural switch. It doesn't allow for that same impulse to be transmitted. So it changes the perception of sweetness. Now, we design molecules based on this theory to have sweet perception. And we can increase the sweet sensation by changing the strength of that aliphatic group. So here's aspartame. So aspartame has our aliphatic group, so there's our phenyl ring. We've got our AH group right here, and we've got our B group right here. And they follow that 5.1 angstroms distancing. Other molecules that give sweetness, amino acids. Certain amino acids that have this configuration can give a sweet perception. Chloroform, if you ever work in a lab, you probably shouldn't put it on your tongue, but chloroform is another compound that's very, very sweet. So remember, we used to use chloroform to put people to sleep for surgery? You would always get a really pleasant, sweet perception before falling asleep. Now when they do it, you get a burning sensation with whatever they use. So carbohydrates are not only important for sweetness, for that detection of sweetness, but it's not just as simple, yes, it's sweet, no, it's not sweet. We can tailor how that sweet profile is released. So is that sweetness really up front? So when you take a bite, do you get overwhelmed with sweetness? Or does sweetness come in and linger? Does it come in after you swallowed that food? And it's a very slow response. So we can actually change and tailor how that perception is influenced over time. We do it all the time in the confectionery industry. Think about layering candy. So a Skittle even, as rudimentary as a Skittle. You have that candy coating, sometimes it's sour, sometimes it's sweet. If you leave it and you're patient, unlike me, I chew my candy and swallow it quickly. But if you're smart and leave it in your mouth for a long time, you'll notice that sweet perception changes as a function of time. Gobstoppers is another one. Molecular gastronomy, so this whole area of using chemistry and science and physics to engineer unique flavor responses, does this all the time. So we can change when that sweetness profile hits. So imagine consuming a food where it's initially really sour and then gradually it becomes sweet. We can do all kinds of really cool culinology. We can use all kinds of really neat culinology tools to actually tailor that sweet perception. The other challenge with sweetness is it's not linearly responsive. Meaning if you double the amount of sugar, you don't double the sweetness. So that becomes very challenging, especially in consumers where we chronically overconsume sweet products. So there's a whole bunch of literature right now coming out that if you consume diet sodas for a long time, you have a higher prevalence of obesity. Well, a diet soda isn't going to directly cause you to be obese, right? There's no calories in it. But if you're chronically exposed to that really sweet perception, we require a lot more sweetness to get that same perception as though we haven't had sugar in years. So if we're chronically exposed to very high sweet foods, we want to consume even sweeter foods to get that perception of sweetness. So it's extremely complex. The concentration effects, the food structure effects, as well as the different compounds that are actually inducing sweetness all play a role in determining whether or not that food comes across as sweet or not. Now, we can replace sweetness in foods. We could get rid of all the sugar in the world, take it out of every food, and we could still have the desired sweet perception by using synthetic sweeteners and natural things like stevia. We could do that. But what we lose is two really important attributes. The first is the ability to form color. Again, when we talk about color, the Maillard reaction is one of the most important universally used chemical reactions to increase the palatability of foods. Roasting of coffee, fermenting of chocolate, roasting or conching of chocolate, production of maple syrup, toasting of bread, baking of desserts. All of these reactions, or all of these processes, increase the palatability of foods based on the formation of volatile compounds. Again, if you've ever eaten pate versus a seared steak, the flavor profile is extremely different. Remember when we talked about or watched that video on Impossible Burgers? They look at all the different minor components, parts per million, that are formed during the breakdown of that heme group in forming flavor components associated with that meat flavor, that umami flavor. We have to figure out what every one of those molecules are if we want to create a meat substitute that actually tastes like meat, which is what Impossible Burgers is trying to do. So when they create that soy hemoglobin, I can't even say it, hemoglobin or whatever it is, that soy-based hemoglobin, that's essential for creating very minute amounts of chemicals that have a sensory perception, either a flavor or an aroma associated with that. So when you walk into a good steakhouse that has had a slow roast, what's the meat that they always cook for hours? Prime rib. So if you go to a good restaurant that's producing a prime rib, that's the first thing you smell when you walk into that restaurant, is all the byproducts of Maillard reaction and the breakdown of that heme group. So again, when we talk about color, the brown colors of caramels, the brown color of soda pop, all of that is essential when we talk about the importance of sugar in our food. So again, it's not just about sweetness. Now, when we talk about color, we're going to talk about two primary reactions of carbohydrates in forming colors, the two most important. The first being Maillard reaction, which we've already seen the demo for. The second being caramelization. The difference between those two reactions is caramelization doesn't require a reducing sugar. Maillard reaction must be a reducing sugar. So of these sugars, we have three different disaccharides. We have sucrose. So if we look at sucrose, we've got two hydroxyl groups, two oxygen groups within that ring structure. Is this first one a reducing sugar? Is this one able to undergo linearization? So we go one carbon away, does it, oh man, this thing is sensitive. We have one carbon away, does it have a hydroxyl group off of it? No, so this end's not reducing. If we go over here, one carbon away, does it have a hydroxyl group in it? No, it's 2 carbons away. So this sucrose is a non-reducing sugar. Sucrose will not partake in the Maillard reaction. Let's look at lactose. So this is our condensed milk. Remember the demo we did with condensed milk? So if we go here, again, we've got a non-reducing sugar. We go over here, is it a reducing sugar? Yes, lactose is able to undergo the Maillard reaction. So in that creme de leche, that lactose is able to partake in that chemical reaction that undergoes Maillard reaction because the second monosaccharide in that disaccharide is able to form the linear form at specific pHs. If we go down to maltose, again, maltose is a reducing sugar. So the ability of that molecule to undergo linearization determines whether or not it will go into the Maillard reaction or if it won't go into the Maillard reaction. Caramelization, it doesn't matter. Reducing, non-reducing, both of those sugars will undergo caramelization, which we're going to look at on Thursday. Now, 7 minutes, 5 minutes. Let's stop here. We're going to pick up here on Thursday. We're going to talk about caramelization and Maillard reaction and a little bit about texture on Thursday.